Good afternoon, good evening, everybody. Welcome. Hello, and a warm welcome to this webinar where we'll be discussing vaccine related topics. Today, we'll be discussing the topic on vaccine development from lab to clinic and also looking at the landscape beyond the lab. This webinar is part of a series of planned webinars with the goal of providing information on available career options in the field of vaccinology. My name is Caroline Matter, and I am the Vaccinology Workforce Development Program Strategy and Planning Manager at the KEMRI-Wellcome Trust Research Program, together with my colleague, Professor George Warimwe, Miriam Munene, and Lavender Nafula. We welcome you to this session. Before we start, a few housekeeping matters. Kindly note that this webinar is being recorded, and kindly feel free to use the chat option to share your questions or comments during the presentation, and we'll address them during the Q&A session. We will have a few open slots for people to ask directly questions to the presenters. If your internet is stable, you can raise your hand using the available option. As earlier mentioned, this is a series of planned webinars with the goal of providing information on available career options in the field of vaccinology, which is being run under the KEMRI-Wellcome Trust Research Program, a world-renowned research health unit of excellence based in Kilifi and Nairobi in Kenya, as well as Mbale in Uganda. Over the years, we have excelled in the use of novel ideas and collaboration to achieve better health for Africa, while also developing African scientific leaders. It is under this program that we have the Vaccinology Workforce Development Program that aims to expand the technical know-how and capability of sustainable end-to-end vaccine research and development in Africa through training. And we do this through three channels. One, through internships, which are at human vaccine manufacturing institutions that aim at establishing a mentorship scheme that links interns with senior vaccinologists and market systems, and also engagement with academics globally to help them share and strengthen their experiences and in the vaccine research and development and manufacture space, and also help them advance their career development. The second pathway we normally do that is through a fully funded master's level course in vaccinology, which is presented both in English and in French to successful applicants who we currently have shortlisted for the next cohort. And the selection is based on direct involvement with vaccine research and the excellence in professional and academic achievements. The third pathway is through webinars such as this, which targets early career scientists, graduates, and undergraduate university students in Africa and worldwide to be able to provide information through online platforms on possible career options and what that would look like to help in informed career decision making. So this is something that is in alignment with the Africa Union Partnership for African Vaccine Manufacture. And they have an ambition to increase local vaccine production to meet at least 60% of Africa's routine immunization needs by the year 2040, and also strengthen the capacity for vaccine research, discovery, and manufacture in Africa. This webinar series is part of this engagement. Today, we are glad to have amazing speakers, Dr. Pei-Yin and Dr. Erlyani, who will be discussing two related topics, the vaccine development, understanding from lab all the way to the clinic, and also understanding the landscape beyond the lab. So I'll do a brief introduction. Dr. Pei-Yin is leading the early development team at Hilleman Laboratories. She is a trained virologist with more than 10 years of experience in vaccine development. She obtained her PhD from University of Wisconsin-Madison and pursued her postdoctoral training at Wadsworth Center in New York State Department of Health and the University of Wisconsin-Madison, where she gained her training in biosafety level 3. Her industry experience started when she joined Sentinex Therapeutics in Malaysia, where she developed vaccines against enteroviruses. And after successfully transitioning an enterovirus A71 vaccine into phase 1 clinical trial, she joined Singapore's immunology network to develop therapeutic antibodies against viruses. She joined Hilleman Laboratories in 2021 as the senior scientist and is currently the senior manager at Hilleman, leading the early development team that encompasses analytical development and preclinical teams. Our second speaker is Dr. Erlyani Abdi Hamid, who is the head of strategy planning and PR. Her career spans science and academia to strategic corporate leadership in global health. Initially trained as a stem cell biologist, she completed her research in collaboration with the University of Cambridge and works at the Genome Institute of Singapore. She now leads strategic planning and corporate development at Hilleman Laboratories, a company developing affordable vaccines for low and middle income countries to help improve public health outcomes. She also leads strategic initiatives in partnership with charitable foundations and non governmental institutions working to catalyze positive change in global health. Dr. Erlyani is a proud champion of women leadership in science and global health, having previously led a diverse work group empowering women in STEM careers and driving dialogues with women leaders in global health. I would like to welcome both Dr. Pei-Yin and Dr. Erlyani to do their presentation. Over to you.
Thank you, Caroline. Let me share my slides. Can you hear me okay? Is it clear? Can you hear me? Oh.
Yes, we can hear you.
Okay, good. That's good. Okay. There you go. Okay, so the title of my talk today is Vaccine Development from Lab to Clinic. So I would like to first share with you that vaccine development is a lengthy and costly process, and success is not certain. So development of a new vaccine can take up to 20 years and cost up to 500 million USD and $1 million. A lot of the vaccine can enter pre-discovery and preclinical phases, but only a few will be approved. So to simplify the process, I'm breaking down vaccine development into 6 steps: discovery, preclinical, clinical, regulatory approval, manufacturing, and quality control. So for discovery, during the discovery phase, it is crucial for us to understand the pathogenicity and identify the vaccine candidate. This is followed by the preclinical stage where we characterize the vaccine, develop the process to make the vaccine and purify the vaccine, formulation, perform toxicology studies, and develop assays. After completion of the preclinical stage, there will be an IND submission. Upon approval of the IND document, we will enter clinical phases, and there are usually 3 phases of clinical trials. Phase 1 involves safety and tolerability. Phase 2 involves more volunteers. During the phase 2 clinical studies, generally the investigators will look at safety and dose ranging of the vaccine. And during phase 3, the investigators will look at the vaccine efficacy and also compare lot-to-lot consistencies. Upon completion of all 3 clinical phases, the investigators will have to interact with the regulators and submit a common technical document for licensure. And during the manufacturing phase is when there is a large-scale production of the vaccine, and the vaccine is tested to ensure safety and potency. And finally, quality control is crucial, and quality control is involved in a lot of these phases, and this is important to monitor the safety and quality of the vaccine. So for the purpose of today's talk, we will be focusing on the first 2 phases, the clinical and the preclinical stage. So if we dissect the discovery and the preclinical stages, it involves many steps. First of all, it would be identification of pathogen. Once we have identified what pathogen of interest we're interested in, Then we will have to identify the antigen on the pathogen that could elicit protective immune response. After identifying the antigen, we will have to decide on vaccine modality, decide on what vaccine modality will we use to develop this vaccine. And after this, we'll have to perform proof of concept by using animals to demonstrate that the vaccine can elicit immune response and also protective response. Once we have performed proof of concept in vivo, we will optimize the vaccine by further modifying it, adding different formulations and excipients. After this, it involves process development where we scale up the production and purification of the vaccine and develop assays to analyze the vaccine. And finally, non-clinical studies will be conducted to demonstrate the safety of the vaccine, the biodistribution and efficacy of the vaccine. So now let's look into identification of pathogen and antigen. While we're selecting the pathogen of interest, there are a series of questions that we need to ask ourselves. First of all, what is the pathogen? What is the pathogen of interest? What is the structure and diversity of the pathogen? Is it a bacteria, a virus? Is there any strain variations? Because all of this information could affect antigen selection, which is the next step. So if the bacteria or viruses consist of multiple strains, we may have to consider a multivalent vaccine, meaning a vaccine consisting of multiple components. Second of all, we will have to look at the pathogen life cycle. How does it spread? What does it replicate in the host? So this information affects antigen selection, and it will help us determine vaccination route. So for example, if this is a pathogen that affects the respiratory route, it might be good to use to immunize the vaccine through the nasal route. The third item that we have to look into is the natural immune control and escape of the pathogen. So find out what type of protective immunity is elicited by natural infection. Is it a B cell response or is that a T cell response? Clinical manifestation. Look at the disease caused by the pathogen. Understand the disease burden. How many deaths does the pathogen cause? The demographic of infection. Who will be affected by this pathogen? Is there any specific risk group or age-specific infection rates? Again, this information will affect vaccine design. And very importantly, is there a need for a vaccine? And is there any available candidate vaccine out there that is currently in preclinical or clinical trials? If there's already vaccines in the market, maybe there's no need to develop a new one. Okay, so for selection of candidates, there are multiple approaches. The easiest one would be to look into the literature and find out what has been done. Secondly, it would be to study the protective antibodies elicited by natural infection. And third, it would be to use the conventional approach. Within the conventional approach, one would grow up the pathogen, dissect and purify individual components within the pathogen, perform immunogenicity studies, and then identify the components that elicit protective immune response, clone the genes, and then further characterize the antigen itself. The final approach would be reverse vaccinology approach. This involves whole genomic sequencing of the pathogen. Gene sequencing is currently very popular right now and very common. So after getting the sequence, we could use computer prediction computer to help predict the candidate antigens, prepare the DNA, and then express them into recombinant proteins, perform immunogenicities of these recombinant proteins, and then identify protective antigen. We can then, after identifying the protective antigen, we can further characterize this antigen. So there are pros and cons between conventional and reverse vaccinology. For conventional vaccinology approach, this is only applicable to cultivable microorganisms. If you can grow it, you can use this approach. It is relevant if the antigen of interest is one of the most abundant antigens from the pathogen, and it can also identify structural components of the microorganism. One of the benefits of conventional vaccinology approach is that it can identify non-proteomic antigen as the antigen of interest. So this includes polysaccharides and lipopolysaccharides. For reverse vaccinology, one of the advantage of it is that it could be used for non-cultivable microorganisms. So it can be used to identify all antigen that includes structural and non-structural components. However, it cannot identify non-proteomic antigen as effective antigen. And finally, antigen that are not immunogenic during infection can also be identified using the reverse vaccinology approach. So this is a very good time to introduce the target product profile and its importance. So the TPP is a structured template summarizing crucial information with clear end goals. This is a very dynamic document. You can modify it as you go and as we obtain more information from our experiments. So the key functions of a TPP is to use as a strategic guidance to assist in determining product features. So it can also be used as a regulatory blueprint, a technical document to elaborate the product attributes vital for quality assurance and the associated manufacturing methodologies. This can be used as a due diligence reference to assess product viability and set valuation parameters. The benefit of having a TPP is that it can enhance R&D efficiency. Oftentimes, there are gaps between R&D and commercial sector. And the TPP can use as a very good bridge to bridge these gaps. The other benefit of having a TPP is it can provide positive regulatory outcomes. A lot of time we integrate information in TPP in our NDA, and that leads to fewer requests by the regulators. And a product with a TPP will have a higher success rate, commercial success rate. So here's an example for a TPP for an influenza vaccine. So within a TPP, we will include information such as efficacy, safety, tolerability, the different attributes, the route of administration. One can set minimal acceptance criteria and preferred acceptance criteria. For example, for an influenza vaccine, the minimum acceptance criteria could be an intramuscular injection. As I said before, if this is a pathogen that affects the respiratory tract, it might be a good idea to develop it as a nasal spray. So in addition to these attributes, we would also include information for preclinical studies, the different qualities of the vaccine, the formulation, what kind of clinical studies are we looking at, the market analysis, the target demographic, the competitive analysis, and we could also include information such as which regulators guideline are we going to follow. This depends on where this vaccine will eventually be marketed. So now let's get back to the different stages. Once we have identified the antigen, this is a good time to think about what vaccine modality do we want to use to design our vaccine. There are many different types of vaccine modality, live attenuated, kill whole organism, toxoid, subunit. Subunit includes purified protein, recombinant proteins, polysaccharide, peptides, virus-like particles. One of the most successful virus-like particle vaccine is the human papillomavirus vaccine. In addition, there is also outer membrane vesicle vaccine, polysaccharide, protein polysaccharide conjugate vaccines, viral vector vaccine, nucleic acid vaccine. Nucleic acid vaccine is very common right now because of SARS-CoV-2. So this could include DNA and RNA vaccine. There are two types of vaccine modality that is currently still under experimental stage. That would be the bacterial vector vaccine and antigen presenting cells. So vaccine modalities can affect immunogenicity, tolerability, manufacturing, and analytical complexity. So on the y-axis, on the left-hand side here, we have immunogenicity. On the x, we have tolerability. And on the right side, y-axis, we have manufacturing and analytical complexity. I'm going to give you an example. So for example, a native virus vaccine. This type of vaccine will be highly immunogenic, but it will be low in tolerability. And the manufacturing and analytical aspect will be a little bit more complex compared to something like a recombinant protein vaccine. So a recombinant protein vaccine or a VLP vaccine is on the other spectrum. It is low in immunogenicity, but highly tolerable. And the manufacturing and analytical complexity is relatively low. So when we're choosing vaccine modality, we have to think about the science and the technical aspect of the vaccine. So this is because the pathogen itself will affect what vaccine modality one could choose. So for example, if the target population are immunocompromised individuals, live attenuated vaccine may not be suitable because they are low in tolerability. So once we have identified an antigen will also define vaccine modality. So for example, polysaccharides or conjugate vaccine, if the antigen is a polysaccharide, the vaccine modality will be a conjugate vaccine. So what vaccine modality we choose will subsequently affect the downstream process. So for example, a conjugate vaccine will require an adjuvant to increase its immunogenicity. And the vaccine modality will also affect the process development. If it is a viral vector or an inactivated virus vaccine, then it requires a cell substrate. So while we are thinking about vaccine modality, we also have to think of these three questions. Is this technology patented? If it is, it might not be a good idea for us to pursue this modality, because then we will have to pay, we will have to think about licensing of the technology. The other things that we have to think about is the whether or not GMP grade material is available. And finally, what is the feasibility of scaling up of the production? So once we have identified the vaccine modality, we will produce the vaccine and followed by performing proof of concept in vivo. So to perform proof of concept, this is usually done using animal models because we need to demonstrate the vaccine can elicit protective immune response. There are two different types of animal studies. The immunogenicity studies will determine if the vaccine candidate can provoke an immune response. Usually, mouse model will be used. And we can look at T cell or B cell response by examining the cytokine production and or the presence of specific antibodies. One of the very useful tests will be the neutralization test. Neutralization test will assess whether or not the vaccine elicits functional antibodies that can neutralize the virus or the bacteria. The second type of animal studies are the challenge studies. So in a challenge study, we can determine if the vaccine candidate can protect animals from infection. So different animal models will be used. It depends on the pathogen. The animal model that we use has to be susceptible to the pathogen of interest. Challenge studies may not be feasible because especially when highly lethal pathogens are used. So for example, a biosafety level 4 pathogen, if we're looking at trying to design and develop a vaccine against a biosafety level 4 pathogen, it would be very challenging to perform a challenge studies because we would require a biosafety level 4 environment and lab. So the bottom here, we're showing an example of how a challenge study is being performed. Usually we will vaccinate the animal, and then boost it again with second dose, and finally challenge the animals with the virus or bacteria, and then monitor for survival. Once we have performed proof of concept, this is a time where we can optimize the vaccine. So if we look into a vial of vaccine, in addition to the active ingredients, there are also inactive ingredients. The active ingredients will include the antigen itself, and inactive ingredients could include adjuvant, preservative, and stabilizers. So the goal of the adjuvant is to improve the desired immune response, and this can be a synthetic molecule or a complex biological extract. So ideally, adjuvant themselves will not elicit any immune response. So the graph at the bottom demonstrates that in the absence of adjuvant, the immune response is relatively low. With adjuvant, we can elicit quicker, higher, and more long-lived immune response. And here we have a list of pharmaceutical excipients that are usually used for vaccines. They include surfactants, amino acids, proteins, antioxidants. All of these serve different purposes within the vaccine vials. So now we talk about process development. Process development includes 3 different components: upstream process. The team within upstream process is responsible to optimize the production of the vaccine to increase yield. Downstream process development, their role is to purify the vaccine so that they can meet specific criteria. And finally, analytical development will develop assays to characterize and quantify the vaccine itself, monitor the impurities within the vaccine, and also characterize the process. So the complexity of process development depends on the nature of the antigen and its source. So for example, here we have inactivated bacteria as our vaccine modality. For upstream process, this includes bacterial fermentation. For downstream team will take the bacteria or the supernatant from bacteria and purify and inactivate the bacteria, or generate protein or polysaccharides. For conjugate vaccines, we will use the proteins and polysaccharide here, put it into a chemical reaction to make conjugate vaccines. Inactivated virus or viral vector vaccines, they involve cell culture, expansion of the cell culture and infection of the cell culture with the infected cells of the culture supernatant from the infected cells. We could purify the virus and inactivate the virus. For recombinant protein vaccine, the transformed microbial cells or transfected cells will be expanded. The culture supernatant will be used for purification to generate these the recombinant vaccines. Nucleic acid vaccines. So for upstream process involve expansion of the transformed E. coli. Downstream process involve purification of the plasmid DNA. To make an mRNA vaccine, this plasmid DNA will be used in an enzymatic reaction to generate mRNA. The mRNA is being produced as an antigen. So for analytical development, the primary goal of analytical development is to develop assays to characterize the product and to ensure safety, identities, the identity of the vaccine, the strength of the vaccine, look at the purity, and make sure that the vaccine meets quality, proper quality. Here's an example of how we prepare recombinant protein vaccines. The transformed cells, transfected cells are being expanded to generate drug substance that is crude drug substance, and this crude drug substance is being purified to make pure drug substance that is being formulated and filled into a drug product. So analytical analysis is involved in all of these steps to ensure that the vaccine meets the specific criteria. Finally, the non-clinical studies. Non-clinical safety and toxicology studies are crucial at the end of the process. So one of the main type of studies is the toxicology study. This is when we perform a toxicology study, we will have to follow GLP standards. A basic toxicology study will assess the toxicity of the vaccine in different target organs, the dose dependence, exposure, and whether or not these side effects are reversible. The route of administration that we use in these toxicology study will correspond to the intended use. So if in human we're performing IM, then for toxicology study, we will use an IM route. The dose used for a toxicology study will have to exceed the human dose, Generally, the number of doses will be equal to or more than doses proposed for human. There's a different type of toxicity study, the reproductive and developmental toxicity studies. These studies are performed if the vaccine target pregnant women or women with childbearing potential. Neurovirulence studies is also one of the non-clinical studies. These studies involve direct intracerebral inoculation, and it is generally used for live viral vector or attenuated live virus vaccine. Virus shedding and transmission studies are used to determine if the vaccine shed and whether or not the vaccine can be transmitted from human to human. And finally, biodistribution studies is used to assess distribution and persistence of the viral vector in animals. So after all the non-clinical studies, it is crucial to, this is the time where we manufacture clinical trial materials, the CTM, in a GMP facility. I just like to highlight that this production of CTM requires GMP grade material. It is a very expensive process and it can take up to 4 to 6 months. So vaccine manufacturing is a very complex process. It actually involves 11 processes depending on whether or not it has an inactivation steps and whether or not it is a multivalent or single valent vaccine. So there's also formulation, filling, and freeze drying of the vaccine before it can be packaged. And the vaccine label itself needs to be in accordance with regulatory requirements. This batch release is very crucial. This confirmed that the vaccine has been manufactured and tested in accordance to the correct procedures, and the national regulatory authorities have to give a final authorization before the product can be released and distributed. And finally, the vaccine is transported worldwide, depending on whether or not it requires cold chain or temperature between 2 to 8 degrees. So the take-home message from my talk is that vaccine development is lengthy and costly with no guarantee success. There are different aspects within the discovery and preclinical stages that I would like to highlight. One of it is the importance of TPP. And the second point is that we have to truly think about the science and the technology used while choosing the different vaccine modalities. And CMC development is very complex and it involves upstream, downstream process and analytical development. The non-clinical safety and toxicology studies in animals are crucial to demonstrate safety of the vaccine. And finally, manufacturing of the vaccine is expensive and complex. That's it. Thank you.
Thank you very much. I'll hand over to Dr. Erlyani. Please go ahead.
Thank you, Caroline. Just bring up my slides. Just make sure that you can see it in presentation mode. Please also feel free to let me know if you cannot hear me well. But otherwise, firstly, I will start. And firstly, I want to thank Caroline for the very generous introduction. I just have a couple of disclaimers here. I'm doing this in my capacity as an employee of Hilleman Labs, and the presentation is only meant for informational awareness purposes. I trust that you appreciate it would require many more hours for us to get in depth into some of these areas in vaccine development. So I know you've heard quite a lot.
Erlyani,
yes.
We're seeing your presenter's mode.
Oh, I see. I think I can switch that. Is that better?
Yes.
Yes.
Thank you.
Yes.
Thanks, Pei-Yin, for letting me know. So you've heard a lot from Pei-Yin already about what it takes to take a vaccine through development, hopefully to clinical success. I guess what I'm just trying to say here with this slide, a couple of kind of broad messages, is that if I can put it in short, vaccine development is a team sport. So I think with the audience in this room here today, I guess the message is you don't have to feel like you have to do it all alone, because in the world or ecosystem of vaccine development, there's a lot more than the science and the technical aspects that you have to think through. There, a little bit of what I will touch on is on the business, the funders, the governments, the policies, and the policymakers, your stakeholders in the global health ecosystem, which will enable you to get your vaccine to success. And these represent early partners, early stakeholders that you should start engaging. So again, won't go through, this is what you should have captured from Pei-Yin's earlier conversation or presentation. Hopefully you get the idea in terms of these 5 major steps. Now, as that is being done, I think it's important that people, the team, the technical team that is developing the vaccine also works in parallel with either a sub-team or a support team or some members on your team that also looks at market analysis. I think epidemiology and target population are very intuitive. When you go ahead to choose a vaccine target, it's going to be based on, the epidemiology and whether there is a target population and who you're trying to help. But at the same time, outside of the scientific and clinical considerations, I think Pei-Yin also mentioned this a little bit earlier, things like IP, you want to know that you are working on something that is not yet patented. Otherwise, you will not be able to pursue it freely, or it will come at a cost. Things like competitive landscape and market size growth. I think on the first competitive landscape, Pei-Yin did already mention that if you find that there are other similar targets being pursued, I would say that it's not a hard no, because as you know, in the R&D cycle, a lot of things fall off. So if you feel that you have a promising candidate with a differentiating factor, perhaps it's something you might still consider. However, it's still just good to know from a competitive landscape what has been done before, what the pitfalls were, and then also use that feedback to feedback into the design of what you're doing in terms of the vaccine product. For commercial companies, market size and growth are going to be very important. For Hilleman, I'll cover this hopefully towards the end. We are primarily concerned with the LMIC markets, the low and middle income markets. And as you know, these tend to be regions that are underserved by the big pharmaceutical companies just because profit margins are not clear. So it depends again on the nature of your organization and where their priorities or what their mission is, to give you an indication of what market size and growth one should go after. Regulatory pathway, I think this was briefly covered by Pei-Yin, and also intellectual property, as mentioned earlier, that could come at a cost if you're working on something that is already patented. Now, I want to cover a few things in the next couple of slides on the commercial principles. Again, these are things that as vaccine developers, if you're a scientist or a technical team member, is a good to know. I don't expect that everyone here is going to be experts in everything that we're going to touch on in this presentation. But I think the one thing that you should try and understand about the vaccines market is that it is quite different. It's very niche and quite different from, in general, the pharmaceutical market. Just because if you think about the product, vaccine is a prophylactic. And so you're trying to create market demand for a product for patients or people who are not sick, right? Oftentimes it's being thought of as insurance, meaning that you take a vaccine so that you don't fall sick later on. And this is important because when you look at a country's economy, the cost of disease, the burden of disease can be quite high from an economic point of view. And so your primary stakeholder or your customers in that sense is going to be governments, because it's going to be the government that bears this eventual cost if diseases are not prevented or tackled early. And that's why the vaccine market is considered very niche. So there are these things called government procurements. So while there are organizations like Hilleman that take a very humanitarian view, we in the end have to work. We are an R&D organization. So to give you an example, we in the end have to work with a large vaccine manufacturer whose priorities are largely going to be commercial. And so they need to know that there is going to be market demand for the product that they're going to make at scale, right? Because they're going to do this at high volumes. So the idea here is to work with procurement bodies to ensure that there is this thing called advanced market commitment. And so before a large manufacturer goes out to, produce millions of doses, they typically already have agreements with various buyers that happen to be or tend to be either your procurement bodies or governments to say that they will procure X number of doses or millions of doses. And that's what you call your guaranteed buyback or pledge and purchase. I think some of the other considerations around pricing and reimbursement also is to understand whether the markets that you're going into, the products that you're developing can be covered under insurance or any kind of healthcare plan. And this can influence your pricing and accessibility to your product. Now, I put here also the marketing and distribution as a principle that you should consider because it's about getting information out there. Now, if we talk about a highly developed, well-connected country or market, this can be fairly straightforward. But, when we talk about the One Health approach, we're also talking about reaching communities that can be quite scattered, that can be have very rural infrastructure. And it's about generating awareness, the significance of the product and why it's important to have something like that in your community and make sure your population is covered. Right. So the important thing here is making sure that the general population understand that your product is safe, who should take it, how many doses, and who's a trusted source for them when it comes to implementation and rollout of the immunization program. And when I say scattered communities and remote areas, I think we also have to remember the behavioral aspect or the social science of it, which is different communities are going to have their own set of beliefs and they'll have a perception of something that is being imposed. And these were lessons that were learned from the COVID outbreak, which, as you know, was global, and there were difficulties for different governments in getting immunization to different rural communities. Right, so moving on. The next thing, the couple things on this slide, I think, is more relevant for large organizations, large commercial organizations. Now, if you come from R&D organization, it's again good to just be aware that these are things that the activities that will continue to take place even after the product has gone out, because it will again feed back into your understanding of how the product works and whether or not there's opportunity to improve the product, and in fact, maybe even the consideration for subsequent uptake in terms of booster shots, as we saw with our COVID-19 vaccines, right? Now, the first one under surveillance, once a product goes onto the market, there's still continued monitoring or continuous monitoring for safety, efficacy, and any adverse events. Depending on where your jurisdiction is, what regulatory body you fall under, they may require additional studies or risk management plans based on the results of this surveillance. I mentioned earlier, potential to update your vaccine, especially when it comes to mutating viruses, and in general commercial terms, they call this life cycle management. And I already mentioned the booster programs. For large corporations, return on investment is going to be a key motivator. Again, this may come at odds with, making sure that underserved markets, unmet medical needs are addressed. Because when you look at return on investment, we know that R&D is high risk, it's costly. And so when they map out, when they create that roadmap for a target product, they need to do estimations on revenue projection, look at the timelines for development, again, look at the probabilities of success, and evaluate the expected ROI over time. Oftentimes, there are going to be products that don't make it pass or make it based on this criteria just because they don't think the market is mature enough or big enough for them to find it a worthwhile investment. Okay, so on the next bit, now that was the commercial principles that we covered. I think now we're moving on to the ethical, social, and policy considerations. So you'll find that when I title my slides "Landscape Beyond the Lab," I'm really talking about beyond the science. I mean, to a certain extent, we are talking about the social sciences, but also looking at how other elements are going to impact the success of the vaccine rollout. From, of course, a clinical perspective, but also from a population perspective. So number one, for us at Hilleman, and I mentioned earlier that I would get to it, is that we look at vaccine development from the perspective of equitable access. So I can give an example. When we make certain vaccines, we want to make sure that it's going to help the right populations. And so we think about what the infrastructure that is available, and I'll cover infrastructure in the next slide, but also in terms of how we can give the product certain attributes that make it more accessible. Therefore, what I mean is makes it easier to ship. If we can make a more thermostable product, it will be less reliant on cold chain logistics, because as I mentioned earlier, when we're talking about scattered communities in remote areas, something like cold chain is not going to be immediately apparent. And so we look at improving existing products so that they can be made more accessible to communities that need them. A lot of it also comes in terms of pricing. So when you look at what the global health ecosystem, what the donors and foundations are looking for, they usually want us to be able to develop vaccine products that are something like $1 a vial or even less. And this is because we need to make sure that we are able to distribute to especially in the low-income market. So tiered pricing strategies are often employed to address this challenge. So you'll find that as you get deeper into the vaccine market economics, you'll find that when governments do a bid for certain products, they tend or end up paying different prices. And in fact, you should be aware of something like Gavi, which is a procurement body that actually negotiates on behalf of a number or coalition of governments in order to kind of represent their interests. And oftentimes these are governments that don't have very high purchasing power, because otherwise these markets or these countries are not going to be able to access those vaccines. I spoke about this briefly earlier in terms of marketing and distribution and getting information out. But I think from a social and policy consideration, especially in light of current times, where even the Community for Childhood Immunization has been terminated in the US. This is a body under the CDC. Public perception. So public trust is critical. You want to make sure that your product is kind of firewalled from misinformation or disinformation. You want to make sure that as you are developing the vaccine, whether you're the scientist or the technical team member, that you are operating with the highest level of ethics and qualities to make sure your product is safe, because this goes a long way in building public trust. Companies should also engage in transparent communication, especially when it comes to vaccine safety and efficacy. It's always important that the public or the population understands, to whatever degree you're able to communicate it, understands your product. What I haven't highlighted here, the nuance, is the communications is going to be critical because That's how they're receiving their information, and that builds towards your public perception. I think vaccine hesitancy has been and will continue to be an area or field of study, regardless of whether or not the US federal funding, is available for it. But education and outreach is going to be important. Nowadays, we all have technology in the palm of our hands, so information gets to everyone in a split second. I think we also, to our, what works against us is the algorithms that are pushing all this information out to the general population. So again, between these 2 points, what is key is that we have very good and clear communications. The infrastructure and health systems really comes into play and becomes crucial when you're talking about implementation and rollout, right? We can develop a close to perfect vaccine, high efficacy, very safe, but at the end of the day, it's the last mile. Even after the logistics and distribution, shipping it out, it really is the last mile. You need a team of healthcare practitioners, to be able to administer the vaccine in your scattered communities, in your remote areas where this becomes essentially quite crucial. And so here I've mentioned a robust and well-coordinated health system. So to make sure that the vaccines that you've developed reach the target population effectively and safely. So beyond, cold chain logistics, which is having the vehicles that can keep the right temperature, you also need mobile units. You also need facilities. You need to be able to set this up. And we need strong health systems, because you need, again, the practitioners of the workforce, people that can handle data management, supply chain, and especially I mentioned earlier the comms. So that's your public health campaigns to ensure that there is good reception to your vaccine. I think I'm doing good on time, and this is in fact my last slide. All of the information that I've shared, and going back to my very first point where, success of a vaccine is really a team sport. We do need to know who the players are in the ecosystem. Oftentimes from an R&D perspective, if we operate in silo, even after going through all of the steps and doing all of your diligence, as Pei-Yin has highlighted in her presentation, you need to be able to have a vision for the product after it leaves your hands. So even for us at Hilleman, when we hand it over to large manufacturing partners, once we've done the R&D and the vaccine development in-house, we actually already have in mind who we're going to engage as a large manufacturing partner, even before we complete the development in our facility. Of course, your stakeholders when it comes to going to different markets are going to be your regulators. Every step of the way, we are also trying to understand what is the market pool. So that's when we look at what different governments are doing, what they are rolling out as immunization programs, what the EPI is saying, and also what do bodies like UNICEF and Gavi, what are they looking for? That gives us an indication of those unmet needs or those demands that are available. We also work very closely with NGOs, foundations, and funders. They tend to be the larger actors that can hopefully help you shift things in the vaccine ecosystem to get your products out. And we always take, as vaccine developers, we always take reference from policymakers. These are the string of acronyms you see at the bottom there. These are different committees that fall under the World Health Organization, and oftentimes they make recommendations on what different populations in different regions would need, and in fact even different demographics. Okay, so I think the last takeaway message here for me is knowing your stakeholders, building trust, aligning your strategies with kind of what they are looking for and what the demand is. Making sure there's transparency in those collaborations and developing strong relationships. So these are things that you should consider outside of the lab in order to try and get your vaccine to success. That's it from me. And I believe, Caroline, we go into Q&A now. I've just stopped sharing my screen.
Yes, we do, and they are coming in quick and fast. So I will just put out the question, and either one of you can respond to it.
Wow.
So there's a question from Patrick Comlan. Thank you, Dr. Pei-Yin, for the presentation. I just want to know, one, how do you determine which animal model is most appropriate for a specific pathogen? And the second question is, what ethical considerations are taken into account when designing challenge studies involving lethal pathogens?
Okay, thank you for the question. So the animal models used for different pathogen, a lot of time it is based on what has been published. A lot of studies would have been done to look at pathogenicity of the pathogen, and from there, we will have a sense of what is the appropriate animal models to use. So for the second part of the question, what is the ethical issue when it comes to challenge studies? So it's the same. We follow IACUC, the International Animal Community. They have a set of rules that we follow. You just follow the same rules. That's it. Does that answer the question, or do you have follow-up questions?
Yeah, so is it the same when you're designing studies that involve lethal pathogens? Is the protocol the same?
The protocol is the same, except that the animal studies are done in a when you say lethal, what does that mean? As in biosafety level 3, 4, or lethal can just mean biosafety level 2, but it kills the animal, right? So if it is biosafety level 2 lethal pathogen towards the animal, generally the basic rules for animal studies is that when the animal is sick and more, it's not moving, that's when we have to euthanize the animals. Or when they have lost more than 20% of their weight, we have to euthanize the animals because of humane reasons. But if we're talking about lethal pathogen that involves biosafety level 2 and 3, 3 and 4, so we will need specific trainings to perform and facility to perform those animal studies. And there are not a lot of labs in the world that can that allows people to perform animal studies, animal challenge studies in a biosafety level 3 or 4 labs.
Okay. There's a question from Ruth Rotich. What is the expected time frame for development of mRNA vaccines? That is from antigen identification all the way to formulation.
That's a good question. It depends on the technology. And it depends on how quickly you want to get it done. For us, we have an early stage project involving an mRNA vaccine. So when we chose that project, our antigen is very clear. It's known what the antigen is. That shortened the amount of time. So we were able to perform proof of concept within 3 months. So that's in vivo challenge studies. But it also depends on whether or not you're using a new LNP formulation. If you are, then it's going to take more time. But if you're using LNP formulation that the existing ones, such as the Pfizer BioNTech or Moderna, it's going to shorten the amount of time. So it all depends on what type of pathogen, whether or not the antigen is already known, and whether or not you're using novel technology or existing technologies.
There's a very interesting question here from Anthony. He's quite curious to know when trying a vaccine for pregnant women, which we know is very important, can you use a pregnant animal model like mice?
So vaccines for pregnant women. So I think at the end of my talk, we talk about toxicology studies. There is one toxicology study called the reproductive toxicology studies. Those are using pregnant mice to demonstrate that there is no lethal effect in the mice.
Okay. There's a question here from Mukanda Kadima, who's very interested in how immunogenicity is being assessed in the preclinical and clinical phases during vaccine development. What about the safety, about those preclinical and clinical phases?
Okay, for preclinical, so if we know the antigen of interest, we can also, generally we use ELISAs to examine antibody response. So if you're talking about assays, those are the assays that we use. For what antibody response are we looking for? If we know what the antigen of interest we're designing the vaccine against, we will be looking for antibodies against that antigen. So the concept is very similar because an ELISA is an ELISA. You can coat the plates with an antigen, come in with serum from animals. So that's during the preclinical studies. Or during clinical studies, the setup is the same, except that you are looking at the presence of specific antibodies in human serum. And of course, the secondary antibody will be different. So for preclinical studies, if we're using mice, the secondary antibody will be specific to mice. And for clinical studies, if we're using human serum, the secondary antibodies will be against human. So, oh, sorry, what is the second question, or did I answer all the questions?
So he has a follow-up question on during biological assessment, do we only use animal models for the pharmacological properties, or a deletion plate or a petri dish model can also be used?
Generally, we have to use animal models for immunogenicity studies. I know that organoids are being developed now, but I don't think that's ready now. So we still have to rely on animal models.
Okay. There's a question for you, Dr. Erlyani, from Reward, who say, thank you for your insightful presentation. What's your take on the use of the New Zealand rabbit for development of a protein-based SARS-CoV-2 vaccine?
Role of manufacturers in compensation to these victims. Typically, I would hold that separately from the product owners. The manufacturers can refer to also your your contract manufacturing organization. So typically the compensation to these victims, if there is grounds, will come from the product owner. So this could be a large pharmaceutical company that could, be something that's based in the US or Europe. Yes, they do have a role in the compensation, but it also has to be properly assessed and litigated. I can't think of any examples right now off the top of my head, but I'm happy to follow up with you separately on email if you're interested.
Great. And then there's a question on vaccine hesitancy from Antidius, who say that vaccine hesitancy has been attributed to presumed complications such as the vaccine-associated paralytic poliomyelitis in polio vaccine. What is the role of manufacturers in compensation for these victims?
I'm sorry, I thought I just answered that question.
Oh.
Were you looking at a different question?
Previously, yeah. Okay. Is there any system that does compensation for these victims? Is there any established system or structure that is in the within the space that we are in the manufacturing space?
Not that I am aware of in terms of a system for them to it's usually reported through your health authorities in different countries. And I think if for a very mature, or litigious, if you want to call it, a market like the US. I know for other types of pharmaceutical products, they can have a class action suit. That tends to be the case, but that's not like a formalized system that usually involves kind of looking at, a legal counsel or legal representative looking at, who the affected victims are, and then they will pursue a suit after the product owner. So I'm keeping that separate from the vaccine manufacturer. I hope that answers the question.
Okay, yeah, yes it does. Thank you. Dr. Pei-Yin, there's a question from Fred Omiti. What is the impact of vaccine washout period during clinical trial phases on routine immunization schedules, effect on immunodulation and immunogenicity?
I'm sorry, can you rephrase that?
I can see his hand up. Maybe I can give him the opportunity to ask it. If you can quickly just unmute and ask this question so he can be able to clarify on what he meant. Fred Omiti? Are you there? Yeah.
Yes, I hope you are getting me. I know I'm on transit, but I was asking what the effect, because during vaccine trials we have washout periods. So my question, what is the effect of the washout periods on vaccine trials, and mostly in effects on impact of routine immunization schedule? And also, how does that one affect the immunogenicity and immune modulation?
I'm very sorry, I
didn't
can't
catch
really hear you at all.
Sorry, we didn't also catch that. I think you're on the road. If you're able to just retext that, if you're not able, because you're not very audible. Okay, as he settles and if he'll come back on, we can move on to Nathaniel, who's asked, thank you for this wonderful presentation. I have one question. Is there a correlation between the type of vaccine, for example, attenuated virus, or mRNA or DNA vaccines, and the route of the infection vaccine? I don't know whether that makes sense. Is there a correlation between the type of vaccine and the route of the injection?
I would say probably no. The route of infection.
I think this is the route of administering the vaccine. Is there a correlation between the type of vaccine and how it gets administered into the body?
No, there is generally no such rule. So for example, if I understand this correctly, the question will be, do you always vaccinate through IM with a viral vector, and do you always vaccinate through mRNA, the nasal route with an mRNA vaccine? If that's how I understand it. If that's the question, then no. There's no correlation within the type of vaccine versus the route of administration. Usually, if we want to administer a vaccine through a specific route, we have to formulate it differently. The same type of vaccine can be formulated differently. So for example, an mRNA vaccine currently, generally they are vaccinated through the intramuscular route, right? If you want to make it a nasal, immunize it using a nasal route, we have to completely change the formulation of the mRNA vaccine. And then only it can be vaccinated through a nasal route.
Okay. And could you please elaborate what determines vaccine modality?
What determines vaccine modality? So the pathogen of interest, the antigen. So for example, if we understand, we know what the antigen is. If it is a protein antigen, then we have a lot more choices. So we can use mRNA vaccine or nucleic acid-based vaccine or recombinant protein vaccine or viral vector vaccine. But if it is a polysaccharide or a lipopolysaccharide is the antigen of interest, then it limits our vaccine modality. We have to go with a conjugate vaccine because we can't use anything else.
Okay.
So the antigen of interest will affect the vaccine modality of choice. The target population will affect vaccine modality of choice. If we are targeting, the vaccine is meant for immunocompromised people, then viral vectors are not a good idea because viral vector will most likely have more immune-compromised people will not have the immunity to tolerate a viral vector.
Okay, thank you for that. Dorcas has asked a question. Hello, thank you for the presentation. I have a question. How can safety signals be monitored across different phases of clinical trials?
Okay, I guess that's a question for me. So when we conduct clinical trials, all of the assays that we use for the clinical trials have to be validated and qualified. So we will be using the same assay, and we'll be using the same reagent to set up the assay during different phases of clinical trials. If in the event where we need to change the assays for monitoring the immune response or safety, oh, sorry, this is safety signals, right? Not immune response. Yeah, sorry. So the safety signals is pretty much the same. There are different levels of, the clinicians will set levels of different safety levels, and these safety levels are exactly the same at different phases. So when patients are administered at different phases of the clinical trials, their observation will be recorded, and there's a chart that the clinician will go through and make sure that they're aligned.
Okay. Ignatius has asked, what adjuvants are being evaluated to enhance the immune response to the recombinant protein antigen, and how do they affect the balance between humoral and cellular immunity?
Okay, so there are many different types of adjuvants that are being used right now. The most common one will be the aluminum salt, not salt, aluminum phosphate. So that's the most common adjuvant. And there are also different types of adjuvant. All of these adjuvants can elicit different, either more towards humoral or cellular immunity. So depending on, again, we look at the natural immune response against the pathogen itself. What is more important? Is it the B cells or is it the T cells? So based on these, the immune response triggered during natural infection, while we are designing our vaccine, we also want to choose adjuvants that are more towards the B cells or the T cells response. So different adjuvants target different humoral and or cellular immune response. These are all published, and it's good to go through them and find out which is better suited for the pathogen of interest.
Okay. Sorry, you have quite a lot of questions. I think this should have been a workshop. There's a question from Evelyn. When you say that you use animals that are susceptible to the pathogen, does that include mice from the wild or only SCID-HU mice? And also, beside mice, are there other animals like monkeys or baboons that can be used since they have similar phylogeneticity with humans?
Okay, usually we would prefer to use wild-type mice. So for immunogenicity studies, wild-type mice is fine. And for challenge studies, sometimes we may have to rely on immunocompromised mice if the wild-type mice are not susceptible to the pathogen and we have no other choices. Yes, we can use other animal models such as rabbits or monkeys, but the cost of large animals is very expensive. That's why, and if you can't really include many more animals if we're using larger animals. First, the cost is expensive. Secondly, we cannot include so many more animals. That's why at the very beginning phase, we will rely more on mice. Once you have shown proof of concept in mice, then we can move on to larger animals. But you don't want to go there until you have proof of concept in smaller animals, or else the cost of running a large animal studies is a lot.
Okay. There's a question here from Michael on resistance. So he says resistance to any disease-causing microorganism is likely to vary across individuals. How do you ensure that resistance to an illness could be attributed to the vaccine and not the individual's immunity when you're conducting challenge studies? Does that make sense?
Sorry, is that
So
a
he's
question for
asking
me?
about resistance. How are you able to ensure or attribute the resistance to the illness based on how do you determine the attributed resistance to an illness to the vaccine and not the individual's natural immunity? Are you able to determine what is caused by what? Is it the individual's natural immunity or is it because of the vaccine that has been introduced into that person?
I'm sorry, can you rephrase that?
So I think it's a case of natural immunity vis-a-vis vaccine induced immunity. Are you able to attribute for this particular person's this particular person has gotten immunity based on the vaccine that they have received rather than natural immunity which they developed whether they would have gotten the vaccine or not?
Okay, so this is in human, right?
Yeah.
Okay, the question would be if someone has been vaccinated and they don't get sick when they're infected, how do we know if it is the vaccine that's protecting it or the person's natural immunity?
Yeah.
So I think it's very difficult to look at individuals per se, but we look at a general population by comparing people who've never been vaccinated and look at whether or not they get sick versus the people who has been vaccinated and whether or not they are protected or not. Yeah, I think it's very difficult to just look at one individual and say if and to determine if the vaccine is protecting it or whether or not the individual has individual immunity is protecting from the infection.
Yeah. Okay.
Yeah.
The question from Abdi Rashid on which critical process parameters have shown great variability on sensitivity during routine monitoring for COVID vaccines. Also, I would like to push this into the next series that we'll be having next week. He's asked to what extent was the manufacturing timeline accelerated and were any process controls or quality risk mitigations adjusted as a result? So I would like to put a pin on that and have him have an in-depth session with the next speaker who will be in next week, Dr. K,
Yeah.
who will be talking about exactly that particular topic. But maybe you can give an attempt at answering him for now.
Okay, what is the critical process parameter shown the greatest variability or sensitivity during routine monitoring for COVID vaccine? Are we talking about manufacturing of COVID vaccine? Is that what he's asking? What are the process parameter that has the greatest variability during production of the vaccine? Is that the question?
Is he here? Abdi Rashid, would you mind asking the question? Let me unmute you. Kindly ask the question. Abdul Rashid, are you here with us? Okay, as we wait for him to get back online.
Okay, never mind. Maybe let me try this. Let's assume that the question is on manufacturing of COVID vaccine. Generally, when the process has been is being conducted by the manufacturer, it's locked down. There's no variability, or there will be no variability. We have a very tight range at every steps during the manufacturing process to prevent deviation. So the manufacturing team has to follow exactly what is written in the protocol for manufacturing, and there are criteria set at every single steps to make sure that there is no variability at every process, every steps during the manufacturing process.
And then we have a question from Wellington. Hello, thank you for the presentation. During vaccine dosage modulation, how do companies determine the cost effectiveness of vaccine and affordability against the optimum dosage with utmost immunogenicity?
So maybe I'll take that one.
Yeah.
If that's okay. So first and foremost, I would say that all of this is going to be driven by the clinical data. In terms of how well the vaccine works, it's really not a cost consideration at that point in time, because you want to make a product that has higher safety and very good levels of efficacy. Otherwise, there's really no point. The acceptance is not going to be great. But from a pricing perspective, and you're talking about cost effectiveness, there are other components, there are other elements that one can look at. If you're doing it before kind of deciding on whether you're working on a vaccine or not, you would do things like economic modeling and affordability analysis, and also health system considerations to see whether the health system has the infrastructure to take it up, because otherwise that's also an additional cost component, although not borne by the manufacturer, but will be a consideration for the different systems or the different regions that might want to take it up. But there are other ways to also look at the cost component that can be in things like packaging. That can be if there's a trade-off between making a product more shelf stable, and so you can keep it for longer, that goes some way into the cost consideration as well. When it comes to packaging, you can talk about different materials. You can talk about, again, there's going to be trade-offs, but you can put multiple doses in a single sort of administration package, whether that's a single vial or anything of that sort. And I think Pei-Yin may be able to chime in here. I think companies do make a decision on the vaccine image, if I understand correctly. For example, there's not a requirement, but largely in LMIC campaigns, they will use a 10-dose OPV vial because it's lower cost, but the trade-off is you risk higher sort of, your vial being open and higher wastage in those vial images. So I hope that answers your question to a certain degree.
Okay, we have the last 3 questions. One is from Michael. You have touched on using immunity-compromised mice in studies. Do you do the same for humans during the final stage of vaccine development?
No, none of the phase clinical trials will use immune-compromised people. That's just ethically not right. Yeah.
Yeah. So what's the alternative? If you have to, what's the equivalent of that? Do you still use normal human beings and extrapolate or model it out?
No, I think okay, there will be different stages when in terms of phase clinical trials. We will always start with healthy young adults. And if the vaccine is also meant for elderlies or the young children, then there will be an age escalation and age de-escalation. So we will during the clinical trials, after we've shown safety in the young healthy adults, we'll move to older adults, the senior, and there will be steps. So if the vaccine is truly required for immunocompromised people, there will also be a step to make sure that it is truly safe in healthy adults before we have to move into immune-compromised people.
Okay, and we are at the tail end. So what strategies are put in place in animal models to determine the best correlate for protection of the vaccine candidates?
Okay, correlates of protection is a very tricky and challenging bit to find out. So, for example, poliovirus, we know what is the correlate of protection, but that is only after a lot of human clinical data that has been put in place, and we will have to look at, at what level of human serum titer do we see protection. So this takes a lot of effort. For influenza, this practice has also occur in human clinical samples. From human clinical samples, we look at the when the human beings get sick, what is the virus, what is the serum titer, and then after determining that cutoff, we will move into animals and figure out if animal does the same thing with the same titer, does animal get protection? But it's a very challenging things to look at.
Okay. I would just like to just wrap this up. We're almost at the top of the 1.5 hour mark. And as you had mentioned earlier, this is part of a series, and we'll be having a part 2 of the series on the 16th of June at 1400 hours East African time. And we'll actually be discussing the topic that Apni Rahman had mentioned earlier on rapid approaches to vaccine development, which will be discussed by Dr. Kay Hansberger, who is the Executive Director of the Vaccine External Development at Merck Sharp and Dohme. So please just look up for the invitation, log in, and let's continue this discussion. I'd like to just thank everybody who has attended this webinar, who would like to hear about your experiences and what else you'd like us to discuss in upcoming forums. I have posted a link on the chat. Kindly just take a few minutes to just give us an update, leave us comments, let us know how your experience was with the webinar, so that we can be able to use the information and feedback to improve the next ones. We would like to say thank you very much and have a lovely day ahead of you. Any last comments from Dr. Erlyani, Dr. Pei-Yin?
Nope, thank you for having me.
Just one. I highly encourage everyone who is here to tune in for Kay's presentation, because I believe she will go through some real world scenarios, and that is invaluable for your learning. Have fun.
Yeah. Thank you for that. Okay, ladies and gentlemen, we've come to the end of our webinar for today. Do you have a good day, and we look forward to more engagements with you. Bye-bye for now.
Bye.